Okay, here's something to think about. What if the very thing designed to protect you could actually have some, well, unpredictable effects inside your body?
Sounds a bit sci-fi, doesn't it?
It does. But today on the deep dive, we're actually looking into some pretty significant questions being raised about mRNA vaccine technology.
Yeah, this is based on some recent discussions, especially around NIH data. We're drawing from insights shared by Dr. Jay Bhattacharya. He's the new NIH director, particularly from a YouTube video featuring him and Dr. John Campbell.
Right. And our goal here for you listening isn't just to skim the surface. We want to dig into the nuances, the potential complexities experts are talking about now.
Exactly. We know you want to be well-informed, but maybe without getting totally bogged down in technical jargon. So this is our attempt at a focused, digestible look at these questions.
And it's timely, too. I mean, we've seen COVID vaccine uptake drop quite a bit in the U.S. Some reports even say they're kind of dead in the mind of the public.
But maybe more central to our deep dive today is what Dr. Bhattacharya reportedly said based on his look at the NIH data about the mRNA tech itself.
Yeah, the source material suggests his analysis confirms some of our worst fears. Strong words.
Definitely. So let's unpack that.
Okay, so the basic theory.
Right.
The Nobel Prize-winning idea was pretty neat, right? A tiny snippet of mRNA gets delivered, usually in your arm.
The plan was it stays there locally.
Your cells read it, make an antigen like a harmless piece of the virus.
And then your immune system learns, okay, this is the bad guy. Simple enough on paper.
But how does that square with what Dr. Bhattacharya's analysis of this NIH data reportedly shows? That seems to be the core issue.
Well, one of the first big points is control. Or rather, the lack of it, specifically over the amount of antigen produced.
You mean we know how much mRNA we inject?
We can control that dose, yes, reasonably well, like measuring ingredients for a cake.
But then what happens inside your cell?
That seems to be the unpredictable part. The amount of antigen, like the spike protein that your cells churn out, well, it reportedly varies. A lot.
The person to person.
Exactly. Some might make just a tiny bit. Others apparently could produce huge amounts, and there's even data suggesting the spike protein can hang around longer than expected in some people.
That really makes you pause, doesn't it? I mean, basic medicine is all about the right dose of the right drug.
Precisely.
So if the actual dose of the antigen being made inside you is so variable, well, Dr. Bhattacharya reportedly questions how regulators can really sign off on that.
It's a fundamental question about predictability and control, isn't it?
Yeah, and that connects to the next point, right? Where this stuff actually goes.
Yes, the biodistribution. The initial theory was localized action right there in the arm muscle.
Stay put.
But it seems that might not be the whole story. These lipid nanoparticles, the little fat bubbles carrying the mRNA, they're incredibly tiny.
Small enough to travel.
Potentially, yes. Small enough that they might not just stay in the muscle. They could get into the bloodstream, maybe through tiny vessels, even with a good injection technique and travel.
And travel where?
Well, the analysis suggests they can end up pretty much all over. Organs like the brain, thyroid, eyes, heart, liver, systemic distribution, basically.
Wow. Okay, that's quite different from just staying in the arm.
It is. And this systemic spread is cited as a potential reason for, well, for harms being seen in many, many body systems, as the source puts it, a wide variety of adverse reactions.
Because if the antigen is being produced in all these different places,
then the immune system might react in those places too, leading to effects far beyond the injection site. Makes sense, logically.
Okay, so we've got unpredictable dose, unpredictable location.
Yeah.
Was the third major point from Dr. Bhattacharya's reported analysis something about frame shifting? Sounds technical.
It is a bit, but let's break it down. Think of the mRNA code like a sentence written in 3-letter words. Codons, they're called.
Okay. Like the cat sat?
Exactly. Your cell's machinery reads those 3-letter chunks in order to build the right protein, the antigen. H-E, cat, sat.
Got it.
Frame shifting is like, imagine your reading glasses slip, and you suddenly start reading from the second letter instead of the first.
So instead of the cat sat, maybe H-E-C, H-E-C.
Precisely. The reading frame shifts, and you get completely different nonsense words, or in the cell, you could get completely different off-target proteins being made, not the intended antigen at all.
Okay. And why is that a specific concern? Just making the wrong protein?
Well, the really significant worry raised, reportedly by Dr. Bhattacharya, is about what kind of wrong proteins might be made, specifically the potential theoretical risk that some could act like prions.
Prions. Okay, remind us what those are, like mad cow disease.
Exactly. Prions are basically misfolded proteins that can cause normally folded proteins nearby to also misfold. It's like a bad influence spreading through your proteins.
A chain reaction.
A chain reaction, yeah. Leading to diseases like Creutzfeldt-Jakob disease in humans. So the concern voiced is whether these frame-shifted, unintended proteins could potentially trigger that kind of misfolding cascade in the body.
That sounds serious.
It does. Now, it's really important to stress, as the source material also notes, there's currently no evidence this is actually happening with mRNA vaccines.
Okay, that's crucial. No current evidence.
Correct. But the theoretical possibility stemming from this lack of control over protein fidelity due to potential frame shifting is something Dr. Bhattacharya reportedly believes warrants serious attention and more investigation before we continue widespread use.
So, wrapping his main points together, it's the lack of precise control over the dose of antigen, where it goes in the body,
Yeah.
and even the exact nature of what's being produced.
That's the core of his reported argument, yes. These uncertainties raise fundamental regulatory and safety questions in his view.
And it led him to that striking comment mentioned in the video, that maybe we need, 2 or 3 more Nobel Prize-winning advances.
Yes, before the mRNA platform is truly, ready and safe for prime time, so to speak.
Which is quite ironic given the existing Nobel Prize for the foundational work.
It highlights that gap, doesn't it, between a brilliant scientific discovery and its potentially complex real-world application on a massive scale.
He seems concerned about pushing ahead despite these unresolved issues.
That appears to be his stance, yeah. Worry about the continued development and rollout when these fundamental controls aren't, in his view, fully established.
So synthesizing all this, what does it really imply for how we think about these vaccines?
Well, I think it underscores just how complex vaccine mechanisms can be, especially with newer technologies that operate at such a fundamental biological level, manipulating our own cellular machinery.
It's not just introducing an antigen, it's introducing instructions.
Exactly. And when you can't perfectly control the outcome of those instructions, the dose, the location, the fidelity, it naturally raises questions. It highlights why ongoing, rigorous safety evaluation and transparency are so critical.
And the regulatory challenge too, right? How do you regulate something with inherent variability?
That's a huge question these findings pose for regulatory bodies worldwide. It challenges traditional paradigms of dose control and predictability.
Okay, so for you listening, trying to get a handle on this,
Uh-huh.
the key takeaways really seem to be these core concerns. 1, we might not precisely control the amount of antigen your body makes.
2, we might not control where in your body it gets made or travels.
And 3, there's a theoretical question about whether the exact intended protein is always produced because of things like frame shifting.
Right. These aren't just minor technicalities. They're pretty central to understanding how this technology works and interacts with our biology.
So the so what for you is, well, understanding these potential issues helps you form a more informed picture of the whole mRNA discussion.
It's not about dismissing the tech outright, not at all. But it is about acknowledging where the science currently stands, the limitations, the unknowns, and why continued research and safety assurance are really, really important.
Which kind of brings us to a final thought, something for you to maybe chew on after this deep dive.
Yeah.
Given these complexities we've discussed, how do we as a society strike that balance between embracing exciting, potentially life-saving new medical tech?
And the absolute need to fully understand it, to have really robust safety measures in place before widespread use.
Exactly. And maybe what role should ongoing research and crucially transparently shared data play in building or maybe rebuilding our confidence in these new medical platforms?
That's the big question, isn't it? Something definitely worth thinking more about. We'd encourage you to keep exploring, look at different perspectives on this.